I am Lau Xiuqing, and now I will proceed the presentation with the importance of the reported structure in the design of drugs for treatment of influenza A. This figure shows influenza A virus. And my part of presentation will be focusing on neuraminidase. For your information, neuraminidase is one of the two major surface glycoproteins found on the influenza virus particles. Its function is to cleave off the terminal sialic acid from the cell surface glycoconjugates, which is the receptor for the other surface protein, hemagglutinin. It is known that inhibition of neuraminidase can effectively delay the release of progeny virions from the surface of infected cells. Therefore, there is a drug development targeting the neuraminidase protein, as it is proven to be a useful target for antiviral therapy because its active site is highly conserved in both amino acid sequence and spatial orientation, as well as it is prominently located on the surface of virion, so it will be easier to be targeted by the neuraminidase inhibitors. So this figure shows the interaction of the neuraminidase active site with sialic acid in the absence of neuraminidase inhibitors. Active site can be divided into 5 subsites, namely from S1 to S5. So S1, which is made up of arginine 118292371, will be able to interact with the carboxylate side chain of sialic acid. O4-hydroxyl can form interaction with residues glutamic acid 119, aspartic acid 151, and glutamic acid 227. Oxygen and nitrogen atoms of the acetamidol form hydrogen bonds with arginine 152, whereas the methyl group lies in a hydrophobic pocket near isoleucine 222 and tryptophan 178. O8 and O9 hydroxyl groups of the glycerol side chain are hydrogen bonded to the residue glutamic acid 276 and 277 to make an important contribution to the overall binding. So by studying well the interaction of the neuraminidase active site to the sialic acid, so there is a development of the neuraminidase inhibitors to act as a competitive inhibitor to stop the neuraminidase activity from interacting with the sialic acid. So there are 4 main examples of neuraminidase inhibitors lead to the drug development. So namely zenamivir, oseltamivir, peramivir, and laninamivir. So for zenamivir, oseltamivir, and peramivir, they are the derivative of the earliest transition state analog of the neuraminidase substrate 2,3-dehydro-2-deoxy N-acetyl-neuraminic acid DANA, whereas laninamivir was produced via a modification of zenamivir. All of them mimic the substrate of neuraminidase, which is the sialic acid, and they can bind to the neuraminidase with higher affinity as compared to the sialic acid. So as we can see from this figure, so the development of these 4 types of inhibitors are actually based on the sialic acid because they have the basic structure similar to the sialic acid. And then those that are highlighted in red are the side chains or functional groups, which is different from the sialic acid. And those side chains which makes the inhibitors to be able to have more interaction and higher binding affinity to the neuraminidase. So for zenamivir, it has a positively charged guanidinol group replacing the hydroxyl group that is linked to C4. So it increased the binding to the neuraminidase approximately 10,000-fold as compared to sialic acid. For oseltamivir, C4OH and glycerol side chain of DANA are replaced by amino group and pentyl ethyl ester group respectively. For peramivir, it is a cyclopentane derivative with a guanidinol group and pentyl ethyl ester group. For your information, cyclopentane is known to exhibit potent and selective inhibitory effects, especially on influenza virus neuraminidase. And for laninamivir, it is actually the modification of zanamivir where methylation occurs on the C7-hydroxyl group of zanamivir. So neuraminidase inhibitor structures are developed based on DANA or designed to mimic its binding to the neuraminidase, but differing structures results in interactions with different amino acid. So all of these inhibitors with the negatively charged carboxylate group are able to form strong hydrogen bonds interaction with the subsite 1 that consists of arginine 118,292,371. Guanidinol group, which is only present in the neuraminidase inhibitors like zanamivir, peramivir, and laninamivir, they can form stable hydrogen bonds and strong electrostatic interactions with the acidic groups of glutamic acid 119, aspartic acid 151, and glutamic acid 227. Glycerol side chain are hydrogen bonded to the residue, glutamic acid 276 and 277, to make an important contribution to the overall binding of neuraminidase to the inhibitors. Oxygen and nitrogen atoms of the acetamidol can form hydrogen bonds with arginine 152, whereas the methyl groups lies in a hydrophobic pocket near isoleucine 222 and tryptophan 178. So as shown in this figure, we can see that neuraminidase inhibitors have more interactions and higher binding affinity to the neuraminidase as compared to the sialic acid. Therefore, the presence of neuraminidase inhibitors will act as competitive inhibitors to the neuraminidase. Neuraminidase inhibitors, which mimic sialic acid structure in its transition state during Na-catalyzed hydrolysis, represent a class of effective anti-influenza drugs. So as I just explained, they are zanamivir, oseltamivir, peramivir, and laninamivir. So for zanamivir, it is mainly delivered through inhalation and was licensed worldwide since 1999. For oseltamivir, it is mainly administered through oral and was licensed worldwide since 1999. For peramivir, it is an intravenous formulation that is developed only for compassionate use and was licensed in few countries like Japan, South Korea, China, and USA since 2014. For laninamivir, which is the modification of zanamivir, was only licensed in Japan since 2010 and is currently in phase 3 clinical trials in USA. As a summary, after budding, virions need to detach from the host cell by viral neuraminidase, which possess the sialidase activity, to cleave off the sialic acid, thus releasing the virion's progenies. So this step is the target of the anti-influenza neuraminidase inhibitors that are used in clinical practice. So in the presence of neuraminidase inhibitors, it can stop the release of viruses from the infected host cells and prevents the new host cells from being infected. Thus, it can suppress the viral population and allow the time for the host immune system to eliminate the virus and eventually control the infection.